All right, welcome everyone to our first Rooted in STEM Talk for 2025. Welcome to everyone online. If you are in person, please sign in for us so we can keep track of who all is here. And if you are online, stick around so that we have the feedback at the end. Wanted to welcome everyone, and especially our guest speaker for today. Super excited to introduce Sarah Vizel Genco. Yes. And her presentation today is Pixels, Patients, and Predictions, which is all about how AI augmented medical imaging is changing diagnosis and cancer treatment. Sarah is a trained biomedical engineer and now manages the application of machine learning and AI algorithms in clinical trials and academic research with a company called Wibm. Her main focus lies in gaining the insights on how data extraction and medical images such as MRI and CT scans can be further used to understand cancer and how to respond to different treatments. When she isn't in researcher mode, Sarah spends her time with her wonderful husband, adorable toddler, and chunky cat. She also enjoys doing gymnastics, despite not being very good at it.
Once upon a time I was good at it, not so much anymore.
So we are super excited to have her talk to everybody today. It's a really cool looking presentation. And if you are online, feel free to put in questions in the chat. I will be looking at that. And with that, we will go ahead and get started because this is your show. Thank you.
Guys. Well, I've really been looking forward to coming out and meeting you all and talking to you a little bit about what I do. I know there's so much buzz out there about what AI is and are they going to replace doctors? And actually, it's not that big of a deal. So AI is a huge umbrella term. It encompasses a lot of things. I'm just going to focus really on a very narrow scope and hopefully demystify a little bit of that for you. So if we go on to the next slide, just a little bit of an outline. I'll just start with actually introducing, myself and who I am, and a little bit about how I got here into this role professionally, a little bit of background and context into the company I work for, and then we're going to get into more of the technical fun stuff. So we're going to talk about software as a medical device. If that's a term you've never seen before, don't worry, we're going to get into it and define it. And then a little bit more about clinical trials. So what are the types of companies that we work with? Big pharma really as bad as everybody says it is. I actually have a lot of faith based on the great people that I've worked with in clinical trials, but talking specifically about like what their goals are when they're running these trials and how using AI applied to medical images helps them to reach those goals. And then a little bit wrapping up about careers in medical technology. So when I was in high school and even in undergrad, like I basically just thought you kind of had two options. It was like, okay, if you like healthcare, you're either going to go to med school or you're going to go the academia route and you're going to work in a lab. Both very noble professions, but I wish somebody had I could go back in time and have someone to talk to me back then and tell me really how many different types of jobs are available. I mean, I might have avoided a painful foray into finance, which we won't talk about too much, but that is part of the journey. So let's go ahead and get started with the next slide. So I kind of compartmentalized my life. I've moved around a bit, as you can see the different country flags. I started off in Canada, which is still its own country. Thank you very much. So that's where I began existing. Nothing super notable beyond that, but it's a very important part of my life. Moved to the US, had a little bit of an adaptation. We moved to North Carolina. Everyone said y'all. I was like, I did not say y'all. Yeah, lo and behold, I ended up starting to say it. And actually a huge chapter of my life, basically up through high school, was I was like that crazy competitive gymnast that was homeschooled and trained 6 or 7 hours a day. My teammates were a lot better than I was. Some went on to an Olympic medal. So my claim to fame was really that I just got to watch them kind of grow and become elite, and that was really fun. I somehow ended up going to Italy for undergrad, where I studied engineering sciences, and I specialized in biomedical engineering. So basically had a combination of mechanical, electrical, and biomedical engineering in undergrad. Then I did an internship in Spain, signed up for a biomaterials like upper level class that was taught in Spanish, despite the fact that I didn't know Spanish. So that was a lesson in adaptability. I survived. I'm still here standing. Overall, it was a pretty good experience as well. And then I moved back. As I mentioned, I kind of accidentally ended up working in finance, got recruited into that, but thankfully I've made my way back to my true passion, which is healthcare and medical technology. So moving on then to a little bit about the company that I work with, and that I'm really happy to work with. So these are just kind of a whole bunch of numbers out there, but to give you context, it's not a huge company. It's not like a McKesson or a Pfizer or something like that. But it's also not one of those like super startups that still work out of like, I don't know, a repurposed janitor closet as a spin out of a university with like 3 employees. We're kind of in the middle. It's what's called a scale up. So it's grown out of the startup phase. It's now scaling up and getting bigger and bigger. So now we're at almost 100 employees. We have several certified medical devices, which basically means that the tools that we developed, we can actually use in standard of care. Basically what that means is like, you go to the doctor, they say, okay, you need to get this test, they order it for you, and they can use that to make a diagnosis. It is a huge uphill climb to get to that point. We'll talk a little bit more about that when we get to the software as a medical device. But that is definitely a huge accomplishment. A lot of companies die before they get there just because of how much research and funding is needed to just get to the point of getting FDA clearance. And the really cool thing about it that came a little full circle for me is it's actually headquartered in Valencia, Spain. So the fact that I had done an internship in Spain, and even though I had been living in the US for probably 7 or 8 years by the time I started at QBeam, it was nice to kind of have like that reconnection to the culture again and have an excuse to get to go to Valencia, which is nice and sunny, and British people like to retire there because it's warm all year round. Even though I'm based here in Dallas, so I work fully remotely. Moving on to the next slide here. Okay. So a bit about what we do. And I know they always say never read straight from the slide, but there's kind of a reason for me doing this. QBeam applies computer vision to medical images to create imaging biomarkers that improve how we study, diagnose, and treat diseases. That's super clear, right? Yeah, okay, one person nodded, but there's a lot of garbage in there. So I'm actually going to, on the next slide, okay, so there's a couple images that pop up. So before talking a little bit more about what we do, I would just like to define each of these terms to give a little bit of context. So those terms being computer vision, medical images, and imaging biomarkers. So first, on the next slide, we're going to identify what is computer vision. So you know how everyone says like a picture's worth 1,000 words, I say a pixel's worth 1,000 data points. So it's essentially you, just like how like we have vision, right? It's not like photons directly hit your brain and you know what these photons mean, right? There's a means of translation that happens, you know, in your eyes and then through the optical nerve, and then that gets converted to signals that your brain can then use to be like, aha, I see that it's sunny outside, or today sadly it's not, but you get what I mean. And computers work in a similar manner. Some common examples you guys might even like use on a day-to-day basis. One is, if we go to one slide, sorry. So like self-driving cars, that's like, in order for them to know where to go, they have to use computer vision, right? They have cameras at different points in the car, and then that gets imported into the computer, like a certain edgy character right now, Elon Musk. I mean, that's what Tesla uses, and they always say that their computers, their cars are not cars, they're robots. That's the line, and essentially like those robots are interpreting continuously what it's seeing around it to be able to know this is where I need to stop, this is where I need to go. Okay, there's a pedestrian, let's not hit that. Something that I use all the time is whenever I'm out and about and I'm like, I wonder what kind of plant this is. I just take out like my Google lens. I don't know, do you guys use that? Where you can just open Google, then you tap it, you take a picture of what it is, and it tells you what kind of like weird plant it is that you shouldn't eat and whatever. So I use that all the time. And then of course there's the famous like facial unlock, which I think iPhone actually did away with. I don't know, that's outside my remit, so I'm not gonna go deeper into that. But all of these applications use computer vision to be able to interpret the world outside of it, turn it into data so that it can do something useful with that information. Okay, so now that we identified, or we talked about computer vision just a little bit, now we're going to talk about medical images. You might be pretty familiar with that, like CT scan, MRI scans. PET is really cool. That's one of my favorites because that's more of like a functional imaging. I'll have a case study that talks a little bit more about that later. Ultrasound, and then even pathology slides, which that's like an actual, biological specimen. My sister-in-law and brother-in-law are pathologists, so they're always talking to me about what they see on those slides. Okay, so pausing there for just a second. There are a lot of different types of medical images, even beyond these. The company I work for specifically focus on CT, MRI, and PET because of the types of diseases we're most interested in studying. So like CT is right now known as one of the best ones for imaging cancer in soft tissues. MRI is a little bit better at imaging cancer specifically in the brain or in the prostate and then the spinal cord. And then PET is kind of like this, I wouldn't say like new because it's been around for a while, but something that's up and coming is getting specific, what they're called tracers that attach to only a specific type of cell. And then if you kind of can stand up, it's a little bright and hard to see, but if you see the spots that light up and they're kind of like red or orange or green, that shows you like that's where these specific tumors are. So that's kind of why it's my favorite, because it kind of is like superpowers your ability to understand where specific types of cells are. Okay, so that's medical imaging. Now I can go on to the third term, imaging biomarkers. So before I talk about imaging biomarkers, a biomarker is basically anything in the body that can be quantified, and then is this like repeatable, accurate, it's observable. So like the most basic ones that you're used to is like heart rate is a good biomarker, blood pressure is a biomarker. And so not only is it something that is measurable and reproducible, but also you should be able to define, are there specific cutoffs? Like a number isn't really useful until you know how to be able to interpret it, right? Like with blood pressure, okay, they're gonna know what is high blood pressure, okay, now that needs to be treated, or oh, the blood pressure is too low, maybe you need like a saline stat because you've lost some blood volume or something like that. So when you talk about imaging biomarkers, similarly, it's something that you need to be able to quantify. There needs to be some agreement as to what these cutoffs mean, and it's based on medical images. So like for my company, it's based on an MRI scan, a CT scan, or a PET scan typically. Okay, now that we've covered the basics, we can go into some specific technology. Software as a medical device. I heard of like software as a service. So now it's like this thing in tech, it's all like software as a, and then like fill in the blank. Medical device, I think to a certain extent is self-explanatory. Most people when they hear medical device, they think of like a pacemaker, or I think technically even Band-Aid is a medical device. And there's different classes, right? Like obviously a pacemaker that you're going to put inside of your heart has to be more tightly regulated and studied and validated than a Band-Aid. But a lot of the same rules that apply to like a physical device that's implanted into somebody also applies to software. And then there's other things that don't apply that actually makes it a little bit easier to get it through the FDA. So specifically, I'm going to talk to you about our device that's so close to FDA clearance called QP Prostate. So I was actually part of the project that was validating this technology with Mass General Brigham. I don't know if any of you guys have heard of Mass General, but it's one of the best hospitals on the planet. The joke is, well, like the acronym like MGH for Mass General Hospital, also the people there call it man's greatest hospital. But it is like it's affiliated with Harvard, they do lots of great research out of there, and it's also a good community hospital. So that's part of why we went with them, because we're like, okay, if they validate it as a third party, the FDA is going to trust what they do because they know that the quality of the research that they do is like pretty top tier. And they have a data science office that was reasonably well equipped to be able to handle our study. Okay, so let's go into what QP prostate actually is and what it does. So, unfortunately, prostate cancer is pretty common. And I know sometimes maybe for some of you guys, might be a little hard to talk about, especially if you have like any family or loved ones that, have had it. One of my good friends, her dad had prostate cancer, so she's been really interested in hearing about our advancements here. But to give you some context about like what is the need behind the technology, because we don't just invent technology because it's cool. I mean, like that's fun to do, but ultimately no technology is going to be adopted unless it's solving a pain point. So really kind of like the pathway that happens is the patient starts off with like a referral. Usually they get like a blood test, maybe as part of their annual workup once they're over a certain age. And there's something called PSA. They look at the level of the PSA. And if that's high, that's a flag of like, ooh, possibly there's a tumor in there somewhere that's creating a lot of this prostate antigen. So then if they confirm like, okay, that's higher, then we think you can't just stop at the blood test. The blood test really is just a gateway, as in like, is this patient good? Okay, we don't need to do any more diagnostic testing. Or if not, yeah, you need to continue on to do additional tests. And that next step is the MRI. And that's where Gibbing's technology fits in. Prostate MRI is notoriously very, very hard to read. So you'll get like one radiologist who sees it and could have a very different answer than what the other radiologist does. It's also pretty time consuming. So, okay, like, hello, pain point, this is something we can solve. We can potentially, make it more reliable, more repeatable, also potentially more efficient as time goes on. Especially as you want to standardize, like it's so frustrating as a patient, if you've ever had the experience or someone in your family goes to one doctor and they say one thing and you're like, let me get a second opinion, and it's like totally different and you're kind of like, okay, now what do I do with this conflicting information? So that's a huge role of AI, is to be able to standardize a lot of this, where it's like kind of more agreed upon standards. That way, like first opinion, second opinion, third opinion, they're going to align a little bit more closely, and it makes it easier for you to know what your decisions are to be able to move next in the pathway. So after the MRI is done, typically what they would do, if the MRI shows, yes, there are some lesions that look suspicious, so they think it's likely to be prostate cancer, they would move on to biopsy, which is basically where you stick a needle in, you get a little piece of the tissue out, and then you actually send it on to a pathologist and they're going to look at it under the slide. There's two types of biopsies in prostate. One is called systematic, where there's effectively just a grid, and they define regions of the prostate gland, and it's just like 12, what they call 12 pores. And then there's targeted biopsy, which is, in my mind, kind of like the cooler one, because it's you're honing in a lot more precisely where you're going to look. And how do you plan targeted biopsy? By and large, you use the MRI as input. So the MRI, the radiologist is going to try to draw out, okay, here's where the potential tumors are. When we plan to do the biopsy, we're going to make sure to biopsy exactly those places instead of kind of going in blind and like a sort of random grid. So that's one of our goals as well. We've been talking with companies that do what's called fusion biopsy, meaning they fuse the MRI images with the ultrasound to then guide the procedure. So that's also something that's in the works. But as a pre-step, obviously, we first need to be in the step of the MRI. Okay, so now that we've talked a little bit about the patient pathway, we can go into what the technology itself actually is. Let's see, we can skip ahead to the next slide and start talking about that. Okay. This is a little bit more of a specialist slide. Actually, next week I'm presenting to a group of the Society of Abdominal Radiology. So this is one of the slides that they are likely going to see. At the far left, which is a little bit hard to see with the lighting, but those are the images, the MRI images that are going to be an input to the algorithm. There's something called like an axial T2 weighted scan and a DWI, diffusion weighted imaging. And then some of those images are going to be using what's called segmentation. So that's basically you're looking at the whole gland, you're drawing the outline. So you're just looking at, oh, that's a lot better. Thank you. You're looking at, okay, this is prostate, this is not. So that's where you kind of see like the color filled in right here. And it's red versus green, just defining like what the regions are as well. And then there's multiple steps that happen along the way. They kind of identify what are possible lesions, and then the algorithm is trained to also know which ones are more likely to actually be what's called clinically significant prostate cancer. So you don't really want to be like biopsying willy-nilly or get like patients worried when they don't need to be worried. because there are a lot of lesions that are more like what's called benign. So like, yes, they're technically a tumor, but they're not really they don't pose much of a risk for the long-term health of the patient. So that's why we have not just detection, but classification. So detection is just like, these look like tumors. Then we have the classification where we do what's called a risk stratification. So here's what's low risk, here's what's medium risk, here's what's high risk. And we really only show the radiologists like the high risk where we have very high certainty that it's going to be clinically significant prostate cancer. And then we have a lower marker that says like, this is a little like on the edge, but you still might want to look at it and consider it. So moving on to the next slide, talking to you guys a little bit about the outputs. So as you guys saw, we get as input, the MRI images, and then we send a couple of the different MRI images back. There's a few that are more interesting for us to look at. This is just a little bit zoomed in of the prostate segmentation. Here you can see a lesion detection bounding box. That's where you draw a box around where we think the lesion is. And this is a contour where it's more specific to the exact drawing of the lesion. What's interesting from a regulatory standpoint is you need a lot more data supporting the contouring because it's more precise. So in our European version of the product, we had that because it reached the level of like the European, I guess, MDR, medical device regulators said like, yep, that's good enough for us. Like your DICE score or whatever, it's localizing the tumor properly. For the FDA, because we really wanted to expedite things through all of the data that we have right now, we're just sticking with the bounding box. But eventually we might get to contouring. But in terms of how the device is going to be used, honestly, bounding box is probably good enough, even if it's not as pretty, let's say, like a contour looks for. In terms of dictating, like what are the two things that the radiologist needs to know? Is there clinically significant prostate cancer? And where should we focus the targeted biopsy? In my opinion, the bounding box accomplishes both of those goals. So that's something always to like remind the engineering team of, because they always want to like get more and more precise and dive into it. I totally understand the passion for that. But at the same time, like resources are limited and we have to use them very thoughtfully. And we always have to remind ourselves like, what's the goal? At the end of the day, it's better patient care. As soon as we're meeting that goal, it's time to say like, okay, where else do we focus to improve patient care and not spend time perfecting something that at the end of the day isn't actually impacting human lives. And this is details probably harder to see. So on the next slide, it goes into a little bit more detail. So honestly, medical IT is super ugly. I'm just going to be honest. One of the previous companies I worked at, we were shocked that they were still like faxing these reports around because the company I was working for before was based out of the UK. And I know we want to think we're like the most technologically advanced here, but that's not the case. They're doing everything electronically like eons before we tend to adopt it. So anyway, these are what reports look like. It's just like basically like white text on a black screen that radiologists do. But it still has like really important information. So in here, we're giving back very specific details, like what are the number of lesions that have been identified? How big are the lesions? What's the risk category that we put it in? Similarly, so this is the lesion findings, and we also have the whole gland segmentation, which calls the volume of the gland. Something that's interesting about that is they use it to calculate something called PSA density. So if you remember, I said they do blood tests to find out what the PSA levels are, but then they calculate the PSA density, so they divide that by the volume of the prostate gland. And that's a way of getting like a little bit more precise to see if they actually need to be alarmed. Because if like guy number one has like a huge prostate and has like these high PSA levels, and another guy has a teeny tiny one with the same PSA levels, he's going to have higher PSA density. You should probably be a bit more worried about the guy with higher PSA density and not just look at the absolute numbers of what the PSA is in the blood test. Okay, so let's see, I'm trying to remember what's the next slide here. Okay, yes, so talking about the radiologist performance. So at the end of the day, I mean, there's a lot of talk about, is AI going to replace radiologists? And also, is this, like, it's really cool that this technology can do what it does, in the short term, it's for sure not gonna replace radiologists. We just want it to augment the people, right? It's like, the radiologist can feel more confident or more efficient in the workflow that they do. So we did like a head-to-head study comparing, so unaided is how the radiologists performed by themselves without the technology, and then aided is how they performed with using QP prostate. And then we calculated the difference. And the clear message that we see here, let's say like the lesion level, is there was an increase. So that's good. However, it's not like mind-blowingly better, like a 4.8% increase, like that's definitely a trend in the right direction. But it's like, wow, we've totally transformed how prostates are assessed. Like our job here is done. Somebody hand us a Nobel Prize. There's clearly more work to do, but it's still encouraging that we know that we're moving the needle in the right direction. Now the question is, as we're about, like it's probably good enough to get the FDA clearance. Is it going to be good enough to convince hospitals and imaging centers to adopt this at scale? That's what remains to be determined. There are several hospitals that are really interested in working with us, and we're going to find out even more, kind of like boots on the ground at the abdominal radiology conference next week. So we're hopeful. Maybe like in a year from now, the landscape of the company I work for is very different. We know in Europe it's being adopted a lot and people seem to really like it. So we're hopeful that will be similar in the US as well. We've had some radiologists say like, I feel like something's missing when I don't have this. Like if he somehow gets like a prostate scan and doesn't have it, it's like, oh, like where's my QP prostate? So that's a good sign. Like once people get used to it, they don't want to be without it. And so lesion level versus a case level, honestly, it's still trending in the right direction. There's not that much more. Basically at the case level, you're just trying to say like, regardless of where the specific lesions are we correctly categorizing a patient into like, yes, they have prostate cancer, no, they don't. So that's also important to determine, make the decision, are they going on to biopsy or not? But the lesion specific one is also determining where they should target the biopsies. All right, so that is our medical device. We may have one more slide on it. Okay, yes, we do. So some of you guys who take statistics might be even more familiar with some of these measures than I am. Everyone's talking about all these kappa functions on some of the calls with MGB, and I was like, oh man, I need to brush up. But ultimately, what this is speaking to is, remember when I said that the radiologists kind of disagree with each other a lot of the time? So we wanted to see, hey, if they use radiologists who use QP prostate, do they agree with each other more than the radiologists who don't? Again, there's a bit of a difference, about a 4% increase. So like, yes, it moves the needle in the right direction. No, it's not particularly mind-blowing. But like, not to hate on anything, I know the engineers have worked incredibly hard, but I also want to be realistic, right? Within our company, we can't talk about paradigm shifts if it's not truly shifting the paradigm. However, it's certainly laying groundwork for a big shift in the future, and we're continuing to work on and improve the performance of the algorithm. So we're definitely hopeful. And prostate is not the only thing that we do. We have brain, we have lung, we have a lot of things coming through the pipeline. All right, on to the next slide here. Okay, I'm gonna pause and take a breath here. Actually, because I'm throwing so much information at you, I worry that if I go through a whole bunch of clinical trials and get to the end, got questions about medical devices might have disappeared. So would you guys be okay with taking like a couple minutes to stop here and just talk about medical device before we move on? Okay, are there any questions? I sure hope so.
In the chat, if you have any questions, please type them out for us.
Good.
How did you, thanks for your presentation. How did you develop that device? Did you have the picture of the MRIs and then the results, negative, positive, and then you train the learning machine to detect those?
Exactly. Yeah, that's actually a really good question in terms of the methodology. So typically what we would do is we have not just like an image, but like you want hundreds of images, maybe even thousands. Medical device development is harder than like, let's say if you're trying to train an algorithm how to recognize a cat versus a dog. I mean, we have so many pictures of cats and dogs. Medical images are a lot harder to get a hold of because it's called siloed. Like this hospital has 50 and that hospital has 200. But yes, you would feed the algorithm, multiple images. Ideally, they're called, like we trained ours on biopsy paired data. So that's what's called our ground truth. So like when you're really determining like, okay, what is reality? What are we comparing it against? We say, okay, like biopsy is the ground truth. So like we can look at how a radiologist might interpret it, but at the end of the day, pathology is the boss. That's the one that's like at the end of the day determines like, yes, this part of the prostate has cancer, or no, it does not. And so we split the data set in two. We have a training data set. So you train the algorithm and then we want to validate it. And you don't want to mix the two because it kind of creates bias and all sorts of things like that. So that's how the model was developed. And then we also went like with Mass General Brigham as a complete third party, showed the algorithm data it's never seen before. And they also used biopsy as ground truth, and then kind of like looked at the performance of radiologists against biopsy, machine against biopsy, and then radiologist plus machine against biopsy.
So I'm thinking by adding more images over years, you're gonna increase the success rate or the correct answer based on what you are training, right?
Most likely, yeah. So we can iterate and continue training it further and further. And like every time we release a new version though, it has to go through some sort of FDA clearance. It could be a minor version change, but that's the kind of thing that's like, it slows progress a little bit. Like instead of having a linear increase, you have like a step change. Because like in the background, you might be training and improving, but it doesn't reach the patient until you get like the FDA clearance, if that makes sense.
Question from the chat, how does AI handle bias in the medical imaging data sets, especially considering variations across different demographics?
Oh, man. Was this from Dr. Park by any chance or somebody who knows medicine? Yeah, that is something that's continuously being addressed. So a lot of what we do is first when we consider what data we're training on is we want to have as much representation in the data set as possible. We also use what we know about a specific, some diseases, there doesn't seem to be a lot of variation from like one ethnic group to another or by age, you know, by sex. But others, like, yeah, there's a significant difference. So we use what we know about the disease to inform our strategy for picking those data sets. And so that's like one of the companies that I've worked with in the past on some projects, Gradient Health. So they're data providers, and that's one of their big things is that they make sure that you can see what the demographic information is when you compile your training data set to then make sure, like, to try to reduce as much as possible the bias that you have. But it is a tricky problem. I think it's far from being like 100% solved. And sometimes we just don't know enough about a specific disease to know how it could present in different demographics as well. But hopefully at least that touched upon how we go about it a little bit. Representation certainly matters. Yes.
Do you ever worry about AI like giving you like false information or like false diagnosis?
Oh yeah, absolutely. That's why we did that validation study, because we wanted to see like, okay, at the end of the day, you're not trying to, I mean, we want it to be perfect. We know that's not very likely. We just want it to at least be better than what we have today. So we know to a certain extent that it's not always going to be right. Like when you saw the sensitivity, it was somewhere like 84, 85%. So that means like 15% of the time, like it's not getting that categorization right. But it's better than what the human does. So I think that's kind of like the issue, and like the line that you have to balance is you do know it could give you an incorrect answer, but so do people. So it's all about trying to move things in the right direction, knowing that you have to accept a little bit of imperfection. But the good point about this is we're not making diagnostic decisions based on the AI alone or the radiologist alone. And that's why together, it's like, have you ever heard of the Swiss cheese model? No, okay, so, but you all know what Swiss cheese is, right? It's like the cheese with a bunch of holes in it. So imagine like, I don't know, you're aiming something from like this side of the room and you don't want it to get to the other side of the room. If you just have one slice of Swiss cheese, like maybe 1 out of 10 of, let's say, like the Nerf darts are gonna find the hole and get through. But if you line up a bunch of different slices, their holes are all in different places. So like the whole block of the slices of cheese is impenetrable or close enough. So that's like the idea with this as well, is like, okay, the machine is slightly imperfect, but so are humans, and if you put them together, maybe they have complementary strengths and weaknesses so that the two of them work together, have much less weaknesses as a system as a whole. So everyone always like, it's always hit or miss whether people have heard of the Swiss cheese model or not, but I like to use that one because it makes sense.
I was curious about where does your software engineers, where are they located? Where do they work? And how do y'all meet, communicate together? Do y'all use Zoom or Microsoft Teams or just a variety of different, I guess, platforms?
Yeah, okay, a very practical question. So we have a international team. In terms of who's based in the US, it's pretty much all like customer facing. Actually, I'm one of like 3 people in the US, so very small US based team. Almost all of the research and development right now goes on in Spain, at the Valencia office. So they go there in person, but we work a lot over Teams. So like I start work at 7 in the morning because there's a time difference, which is kind of cool because I also wrap up earlier in the day. So we just have to be creative about ways to make sure that like information is flowing. I think one of the risks of being like a very remote team, or even worse, like a team that's really in person, but then other parts of the team that are kind of invisible because they're remote, is that information gets siloed. So we really just make sure that we have a lot of like very open lines of communication, very casual over Teams. And then like also periodic meetings where like the whole company gets together. Like every year in September, we get together and have what's called Beacon. And then like regardless of which country you're based in, everyone's in Valencia and everyone gets together. It's like 2 days of sessions. So but we're looking to expand the US. We actually just closed a $50 million Series A investment round. Super exciting for our stage of scale up, which means we're going to have a little more money to work with to really expand our business. And the US is like a huge area of opportunity for us. So I'm excited. Hopefully like by this time next year, I won't just be one of 3, but I'll be one of 30, or who knows what the exact plans are. Yeah, okay. Alrighty. So this is now getting into clinical trials, which is actually the area of the business where I work. So you're probably all a little bit familiar with how clinical trials work. Basically, a company has a new medication. They need to go through a whole series of hoops to actually get approved to be able to give this medication to patients like in real life outside of the context of clinical trials. So they need to go to phase 1, and that's a small group of patients, mainly just like, okay, what is the dose that's tolerable to patients, like the highest dose that's effective, but also safe and doesn't have a ton of like side effects. And then if they get a seal of approval, like, okay, had pretty good results on phase 1, then they move on to phase 2. They have a bigger group of patients, and then their what's called endpoints change a little bit. The endpoints effectively are the goals that they're trying to reach. What are their big research questions that if they like answer those in a positive way, they can get to move on to the next stage. Finally, phase 3 are what's called pivotal trials, because I could literally, it's like it's pivoting like the strategy. It's like the make or break as to whether the FDA is going to say like, yes, you can start like marketing this to patients and their doctors. So part of what we do is to find the patients who benefit most from a specific drug, maximize the likelihood that the drugs are going to get to move from one phase to the next, and to be able to get ultimately to be able to get approval and enable what's called precision medicine, which is cutting edge, but also one of the most challenging use cases, companion diagnostics. Basically, they go in together. It's a diagnostic tool that's like, okay, you have to get this type of diagnosis in order for the insurance company to approve for you to get this medication. Pharma companies sometimes see it as risky to do that because they don't want to limit the number of patients who are eligible. And that's also kind of a challenge that you're always kind of having to balance of, you want, on one hand, you want to stratify to find the patients who are most likely to benefit, but you don't want to do it to the extent that patients who still should be given a shot at this medication are going to be denied. So there's always that kind of balance between like what's interesting to do scientifically and then what's going to be the real world impact for the drug. So I want to go into a little bit of like what the workflow is in terms of kind of like what are the steps we follow with pharmaceutical companies, and then talk about a few case studies. And I'll try not to make anyone late for their classes. That's at the meeting at 2, what's actually pretty cool, we're talking about the future of AI being used in assessing the efficacy of oncology drugs. So that's actually like very topical, because that's kind of what we're talking about here today. And there's like 40 people in that meeting, so unfortunately I couldn't fit. So anyway, so the first step always is we get a whole bunch of images, we need to do quality control. When it comes to data models, the name of the game is like garbage in, garbage out. So you don't want to train a model on like data that's noisy or doesn't make sense. So we're first double checking like, okay, are all the images you sent to us make sense, decent quality? And there's harmonization. So you can kind of see on the very top left grid where there's like different intensities. Some of them are brighter, some of them are darker. And those differences are not physiological differences in the patient's body. That's a difference in what brand of scanner did they use? Was it GE, Philips, or was it a newer model, or was it an older model? And obviously those are that's kind of artifacts that don't really help you know more about what's going on with the patient. So we actually have an algorithm that's called harmonize. You can think of it as like equalize or normalize, so that the differences you see from image to image is purely based on what's going on physiologically in the patient and not due to like any extra stuff, basically. Actually, we're working with a really huge MS data set right now, and the initial results look really promising. So hopefully we'll get to move forward with that with a big pharma company who wants to do more studies on patients with MS and to submit abstracts to a really cool conference. So fingers crossed. I can't tell you more than that because it's confidential, but figure that's like kind of topical. So still want to share that with you. Okay, now that once we've taken care of the images, they're all good quality. Then we go what's called organ and lesion detection. So we're going to detect, okay, what part of this image is lung? What part of this image is liver? And then beyond that, the lesions are like the potential tumors. So we want the algorithm to be able to see and segment out what are the organs and lesions that we want to extract information from. So this comes into the computer vision approach. The radiomics is basically when I said a pixel is worth 1,000 data points. So it's looking at a very specific part of a medical image. Now there's a lot of information that you can take out of it. Like what is the volume of this tumor, or what is the uptake of a specific tracer, meaning like, I don't know, let's say FDG PET. That is basically a type of sugar that's injected into the bloodstream, and the tumors light up because they take in that tracer, which is also radioactive. And so the areas that are really, really bright will have more of what's called uptake. Basically, it means those cancer cells really, really hungry, they have a sweet tooth, and now we can use that to quantify instead of just looking qualitatively on the images, like just how aggressive that tumor is. There's like literally hundreds of features, so I'm not gonna talk about each single one with you guys, but it's a way instead of just qualitatively looking with your eyeballs, which is not to diminish at all what radiologists do, but there is a certain limitation to what you can see with the human eye. And this allows you to quantitatively measure things. And then we can do all sorts of cool stuff. This is a little, probably outside of the scope of what I can cover in 10 minutes. But it basically, if we can take a patient, let's say they're starting a drug trial on day 1, and be able to predict, is this patient going to respond well to this treatment or not, based on models we've created with similar data. Like that's when we're talking about endpoints, prognosis, and prediction. That's effectively what we're trying to do. We're trying to be able to predict the future of this patient and how they'll respond to medication. So that is the pathway. I have a teeny bit of time to talk about some case studies, but I want to leave some time for questions. But I will say, I think you guys have my full name. It's at the beginning of the presentation and also the flyer. So if you have more questions, feel free to just reach out to me on LinkedIn. I'd be more than happy to chat with you guys. Don't just say like, can you be my mentor? But if you have very specific, because of course I'm happy to mentor people, but it's a lot more engaging if you have like very specific questions or like talk about areas of interest that you have, like as a student, you want to do beyond. I'd be more than happy if we run out of time to answer questions today. Okay, so this is a case study, predicting treatment response to immunotherapy from baseline CT scans. So this is almost exactly what I talked about before with the endpoint prediction. basically a super huge biopharma company. If I told you their name, you would for sure recognize it. Again, confidential, can't say it. But they had done a phase 3 study. So think about like literally hundreds of millions of dollars went into discovering what this drug is, refining it, phase 1 trial, phase 2, phase 3, all for it to fail. They didn't reach their goals. They didn't meet their endpoints. They didn't know why. They're like looking at the data and they're like, okay, some of these patients responded really well. Their lung cancer got a whole lot better. And other patients didn't. And when you look at it as a whole, the results were poor enough that they knew like the FDA was not going to approve that drug. So now they're saying like, okay, how can we save it? It's called like a drug salvage program, where it's basically like failed the initial endpoints. Okay, now can we go back and look at the data and like finesse things a bit? So they came to us with basically like no other context, because they wanted to see if essentially if we're legit. Gave us the data set with no other information about like who went on to have progression of disease, which means like bigger tumors, more tumors, who went on to respond well. And then challenged us to be able to do that stratification, to be able to build an algorithm that we think, say like, these are the features that predict, these are the patients who probably went on to do better on treatment, and these are the patients who didn't. We actually managed it, and we showed them, and they were like, wow, super cool. So fortunately that turned into more like ongoing relationship, and they want to do more studies with us. So effectively what they have now as a result of what they did with us is if they want to try again, do a phase 3 trial, now they can better stratify these patients. At screening, they can run the algorithm on these scans and say, okay, this patient, let's put them on treatment because they have all the markers that predict that they would do well. These other patients, maybe we can put them into like the non-experimental arm. So that's going to increase the likelihood of their success, which is exactly what we're going for. Moving on to another type of case study. So this is a big study I'm personally not involved in, but my colleague Anna is. So it's called Chameleon, and it's essentially the ambition is to create the biggest repository of cancer images in all of Europe. And so beyond like the harmonization that we do to normalize the images and whatnot, one of the things is like we actually built the platform to be the backbone to be able to connect all the information from all of across Europe, not just the images, but other important things like, you know, blood tests or other measures that they're going to do for these patients ongoing. So actually, I think they're finishing it in about a month. So that's pretty exciting. We do a lot of grant-based research where we consider this is more like academic, non-commercial, like this isn't for a pharma to productize anything. But it's still for like the greater good of developing the understanding of like how cancer works and how can we treat it. And it's a consortia that like gets so many organizations working together, which is fantastic to see that happening in the medical community. Last case study, I think, that I put up here. This is another one that we're actually actively working on, another large biopharmaceutical company. I met with them at the Precision Medicine World Conference that was last week. It was super cool in Silicon Valley, so felt very techy, and some Nobel Prize winning scientists presented there, got to learn a lot about advancements. But anyway, we met with this company who also happened to be there. And the guy that we talked to, he's in charge of what's called like basically patient leakage along the pathway. So like there's from like the inkling that something is going wrong with the patient to them getting on treatment and staying on treatment if it's needed. These pharmaceutical companies are concerned with patients who drop off along the way. Maybe they got a false negative on a test, or, you know, maybe some part along the pathway, they just kind of like weren't really sure if it was the right diagnosis or not. And from a financial perspective, they want as many people who should be on the drug to get on the drug. I know it gets kind of complicated to mix finances with medicine, certainly. But that's what these pharma companies are interested in. Something with axial spondyloarthritis, which my great uncle actually has and complains about it. Lots of back pain, unfortunately. It takes a long time to diagnose this. A lot of patients, it takes them like from when they start having back pain and they're symptomatic and they're starting to ask their general practitioner, like, okay, what's going on, until they're actually diagnosed with 7 years. That's a pretty long time to be getting different tests and not knowing what's going on, but being like, I know I have pain, but they're telling me it's nothing. And the reason is because there's not really a sensitive enough test to pick up the earlier stages of disease. So it only picks it up when it's like pretty advanced. So we've developed an algorithm to be able to diagnose these patients much, much earlier using medical imaging. That's like a win-win-win situation, because that's way better for patients because they're actually getting treated, and like hopefully out of pain a lot sooner. It's great for the pharmaceutical company because patients are getting on the drug sooner. So they're certainly interested in that. And then it's like a win for us because it's like more proof that you can use like non-invasive technology to diagnose things earlier. All right. Sorry, I knew I should have brought a water bottle. But that's a wrap for the main part of the presentation. And I just wanted to touch upon the careers part. I kind of mentioned it earlier on, which is there's so much you can do in healthcare beyond med school and beyond academic research. If you are like, I know I want to do something with science, I know I want to do something in health, I'm not sure if either of those two options are for me. There's a huge amount that you can do within engineering, and you don't have to be an engineer. You can just have like a biology degree and you can be involved in a company like mine, managing these research projects, helping to write proposals, helping to, you know, be in business development. There's a lot of interesting stuff, even quality assurance, which is super necessary. It's personally not my thing, but huge variety of jobs that you can do. If you're interested in saying like, okay, hey, this looks really cool, go ahead and reach out to me. I'd be more than happy to continue chatting with you about kind of like more what my path is and what I might recommend given your research interests or work interests. Terrible timing for me to like, yes, thank you. Ooh. And that's a wrap. I know we don't have tons of time for questions. We do have a few minutes.
Yes, real quick, before we get into the Q&A, feedback form for everyone, please. I'm also putting the link directly in the chat. We very much appreciate that.
Thank you so much.
Really appreciate all of your feedback, helps us continue to do this. And if you have questions, please post them in the chat.
So your the harmonization technique, I was wondering, how do you get those to all be standardized? If, for example, you have something that's really high quality and something that either is dark or it's fuzzy, do you have to, in order for them to get like the same like cohesive agreement in your data, like do you have to decrease the quality of the high quality one in order for them to match? Because I'm assuming you can't increase the quality of the other one, but maybe that's
a really thoughtful question. And actually, we don't have to decrease the quality. So what we do is we select what's called the reference group. So we look at, okay, what are the highest quality images of this data set? How do we bring the rest of the data set up to that quality level? So because yeah, most people would have that question. It's like, okay, are you just averaging it and kind of like bringing the best ones down, but the worst ones up? No, we can basically build out that reference group. Yeah.
I know you said clinical trials have been 3 stages. How long can a company, is the general time frame it would take?
Ooh, good question, man. And I've seen it on so many slides. It's definitely, I think the typical is about 7 years once it's gone through like the preclinical stage. So like one pharmaceutical company that I'm meeting with tomorrow morning, they're interested in the PSMA PET, which is so cool. And I'm sad we didn't have time to go into that. The one trial that they're talking to us about is phase 1, and it's a 6-year trial, like just the phase 1. So it can be pretty lengthy. So that's why they want it to be successful, because they invest a lot of time, blood, sweat, tears, and dollars into that whole process.
So check.
Oh, sure.
So I am pursuing a PhD in health data science. This is where the bias question came from. When I finished Dallas College, what are some key research gaps or unexplored topics in AI-driven diagnostics?
Whoa, very good question. That would take me a little bit to think about what are research gaps. I mean, there's certainly gaps in certain areas of diagnosis. I think it depends on if you're trying to do like original research or if you're trying to build on existing original research to create a medical device. There's a lot of interest in looking at assessing metabolic disease with imaging. I think that's actually reasonably far along, but there's a lot of interest. Another huge area outside of imaging is actually on genomics. Like every precision medicine conference I go to or the oncology conference, they're really talking a lot about genomics because like they're basically able to like get a sample of the tumor, do genetic testing, and be able to see what the mutation is. But a lot of those startups seem to get a lot of excitement and then fail. So I don't know a lot about that area, but I'd be really curious to understand why is that failing and if there's some gaps that need to be addressed. But that's a good question. I feel like I didn't quite do justice to it. So if you want to reach out to me on LinkedIn, I might think about that more and we can chat.
So you mentioned about that, the precision, medicine, conference. So did they cover a variety of, I guess, of treatments of, not only oncology, but also immunology and just a plethora of, I guess, other fields per se?
Yeah, that was what was really nice about that conference, because most of the conferences I go to are like very specialized. Next week is Society of Abdominal Radiology. That's like more of a narrow scope. But yeah, this one talked a lot about immunology, but specifically applying immuno-oncology. They talked about some rare genetic diseases as well. I actually cried during one of the presentations because they showed a girl who was fully wheelchair bound, was not able to be like move independently at all. And like literally 2 weeks later, after getting that genetic therapy, was kind of like walking with some support, but pretty wobbly. And then a month after that, she was walking on her own, but like kind of wobbly. And then a year, she was like running down the hallway and just being a normal kid. And it's just like when you get to see like, okay, genetic therapy works. I mean, I get goosebumps every time I talk about it because it's sometimes you get into a moment too where you're like, okay, is all the work we're doing really worth it? Because you see like not great success rates in some trials, but like then you see something like that and you realize it's keep going. So yeah, they covered a pretty wide variety of areas and even like just a lot of like, what is the foundational technology? Like the CEO of NVIDIA, which I don't know if you guys have heard of NVIDIA, but like they develop like all the GPUs. They initially developed it for making computer games faster, but then realized there's a lot of opportunities in other areas as well. So like what's the foundational technology that makes OpenAI and things like that, or like DeepSeek, which everyone was kind of like, ooh, about at the conference. Yeah, so quite a variety.
It is 2:00. Thank you so much.
Thanks, everyone.
All right, that is a wrap.
Questions, guys?